This video shows how asexual and sexual reproduction has historically led to improved food crops. There are over 20,000 species of edible plants in the world, and humans regularly use around 7,000 for food. Over the past 9,000 to 11,000 years, humans have started to domesticate wild plants to use them for food and other uses. Wild plants are plants that grow in nature without the aid of humans. There are very few wild plants that are now used as food sources. Most plants that are used as food sources are completely or mostly completely domesticated by humans. As you can see, the difference between many years ago and the plants we now grow is almost completely different because of the aid of asexual and sexual reproduction. So domestication involved skillful breeding. Breeders used natural variation and the variation from sexual reproduction, and they either asexually or sexually improved the food crops further. The domestication led to great phenotypic changes, where the phenotype is the physical characteristics of the plant. So there was great changes in the physical characteristics of the plant, as well as the altered genotype, so the actual DNA of the plant, which allowed for improved food crops. Domestication also allowed for new varieties of plants to be developed, and most of all, our principal food crops come from domesticated varieties, so things such as wheat, maize, and rice. Asexual reproduction specifically improved food crops in a number of ways. Asexual reproduction refers to all the modes of reproduction of plants where normal gamete formation and normal fertilization doesn't take place. So some examples of asexual reproduction could be division, grafting, using storage organs, and cuttings. So if we look at grafting, grafting is where you cut one plant and you graft it, or you grow it together with another part of another plant, and it forms one new plant with two or more different characteristics from the original plants. You can also use cutting, where you simply cut the plant and replant it. You can do that for roots as well as for stems. You cut where the meristematic tissue is, so that's the tissue that's unspecialized, which will allow it to grow. Grafting is, as I said, a technique whereby tissues from one plant are inserted into those of another so that the two sets join together. So the one plant would be selected for its roots, and we call this the rootstock. So it may have something desirable about it, such as resistance to pests or resistance to diseases, or able to grow in difficult soil conditions. The other plant is chosen for its superior fruit, so it might have a larger yield, and we call this the scion. Obviously, in grafting, there's no actual genetic engineering. The farmers or the person doing the grafting doesn't actually go and change the DNA of the plant, which allows it to be quite a quick process. These are just some ways that asexual reproduction has occurred on plants. The basic feature of asexual reproduction is that there's genetic stability. So that means that there's no variation in the offspring. It's continuously the same and the same. The only thing that could cause genetic variation would be a mutation. To form the initial superior trait, often that comes about because of a mutation. For example, the mutation could cause a plant to have to produce fruit all year round instead of just in the summer or in the winter, or to produce greater fruits, or to be resistant to certain diseases, and so on. And then what farmers will do is they'll reproduce these plants asexually to ensure that more plants with a superior trait are grown. And it's beneficial to do it asexually because that guarantees that the plant will have that the offspring plant will have the genetic trait, the superior trait, because the offspring is genetically identical to the parent. With repeated selection and elimination of the plants that don't have the superior trait, the food crop will eventually consist of plants that only have the superior traits. So there'll be no plants with an inferior trait. If we now look at sexual reproduction, plants originating from sexual reproduction were often different from their parents and from each other. And that's quite an obvious statement. You're different from your parents and from your siblings because of sexual reproduction and the genetic variation that comes with that. Some of these differences between the parents and the offspring are beneficial traits that improve the food crop. These are something like larger yields, larger seeds, larger tubers, larger fruit, resistance to pests or disease, or being able to grow in nutrient-poor soils. Farmers or breeders will select the plants and breed those seeds from the superior plants, while they eliminate the seeds from the plants that don't have the beneficial traits. So, for example, the farmer may take a mildew-resistant plant and breed it with a high-yielding mildew-susceptible plant. So the beneficial trait here is obviously the resistance to mildew. The beneficial trait here is the high yield, but obviously this mildew-susceptible plant is not a beneficial trait. Then the farmer is able to produce a range of seeds in the offspring, and from those offspring, there may be some plants that have a high yield and mildew resistance, so both the beneficial traits. Then he will continuously grow these. He might even start reproducing these plants by asexual reproduction. The plants that are susceptible to mildew or have a lower yield, he will eliminate, and he won't keep growing. And we call this plant a cultivar, which means it's from the same species, obviously, as these plants, but it has slightly different genetic traits. How can plant breeders use asexual reproduction and engineering techniques to benefit food production to help solve the current food crisis? So we know that currently the world is in a food crisis. The world needs to produce at least 50% more food to feed the estimated 9 billion people by 2050. That's 30 years away. That's not very long. Climate change could cut crop yields by more than 25%. The land available for crop production is limited and is continuously being used up. And we need to develop a way to have stress-tolerant crops. So unless we find a way to change how we grow our crops, food security, and especially for the people living in the poorest parts of the world, is at risk. So we can use asexual reproduction or vegetative propagation and genetic engineering to help solve this problem. Some of the challenges faced by plant breeders in terms of future food security is that international development agencies believe that breeding new crop varieties is extremely important for ensuring food security. The new varieties need to be even higher yielding at present. They need to be resistant to pests and diseases. They need to be drought resistant or regionally adapted to different environments and growing conditions so that they can grow all over the world. And they need to be able to use soil nutrients more efficiently. So these would be the challenges faced by these future plant breeders. If we just look at the terminology, propagation is a really important word that we're going to need to know. To propagate means to transmit or spread from one generation to another, with, in this case, usually in terms of hereditary characteristics is what we are transmitting. To produce a new plant using a parent plant, to grow a new plant from some part of an existing plant, both of those could be propagation. A plant that's produced in this way is genetically identical to the original plant. It's called a clone, which is asexual reproduction, and of course there's no gametes. These are all meristematic tissue, and the bumps are auxiliary buds where parts of the plant may grow out of. And we can see they've been harvested from here. Then they've then been planted and they grow, and they're genetically identical because they're all from the same plant. Some cultivars are plants such as cultivated bananas, seedless grapes, and navel oranges produce no viable seeds. So these are plants that humans have genetically engineered to the point that they no longer produce seeds, which means that vegetative reproduction is the only way to propagate them and to form new plants. So now if we look at micro-propagation, also called tissue culture, this is propagation, but obviously on a micro level. This is a process whereby a small amount of plant tissue is cultured in a growing medium to produce a callus and then plantlets. A callus is just a shapeless clump of cells. They're stem cells for plants, so these cells are undifferentiated, and we don't use the whole part of a plant. We just use a little version. We just use a section of the plant to create these calluses. So this is asexual reproduction. Obviously, there's no gametes involved, but the offspring aren't going to be genetically identical to the mother or to the parent plant, simply because during micropropagation, genetic engineering will usually, not always, but usually occur. So plant tissue culture or micropropagation is when the generation of entire plants is formed from a single cell or just a few cells of the plant. It's based on the principle of totipotency. So what totipotency is that within a plant, every single cell in the plant has the potential to develop into any other type of plant cell because it contains the entire plant genome in every cell in its active form. That's in its active form is the important part. So in humans, we know that all our cells do also contain the entire active genome in our cells. Because we develop from stem cells and our cells will eventually specialize into different types of cells. The difference between humans and plants is that in human cells, while we contain the entire human genome, the cells have, and this genome is not in its active form. So the cells can't unspecialize and then respecialize into something else. In plants, they can. So a leaf cell, for example, can despecialize and then respecialize into, for example, a petal cell, just because it has the entire plant genome in every cell in its active form. That's what we call totipotency. So the leaf cell can be converted into a root cell or even a flower cell if it's manipulated by scientists correctly. So scientists are in control of this. This is genetic engineering. The potential of this information is that existing differentiated cells, so cells that have already specialized, can be manipulated to become embryo cells again, so unspecialized, and then they can differentiate back into a new type of cell to divide and grow an entire new plant. So how we do plant tissue culturing or micropropagation? There must, of course, be a completely sterile environment, no microorganisms, no viruses or bacteria. Within this environment, we choose a suitable plant specimen, so the one with desirable characteristics, for example, a high yield or resistance to a certain pesticide. It is then cut into a number of tiny, tiny pieces called explants. You can use any plant part of the plant because of this thing that we talked about called totipotency. These explants are then meticulously sterilized to kill any fungal spores or bacteria endospores that may be on or in the tissue. If it wasn't sterilized, this fungal spores or bacterial endospores would grow very, very rapidly, and the plant would not be able to grow properly. The sterilized explants are then placed in a sterile agar medium to grow. Agar just means nutrient medium. This medium contains a specific mixture of plant hormones, one of the most important ones being auxin, and this encourages de-differentiation of the plant cells. So all the specialized organelles and structures will disappear, and only the general structure of the plant cell remains. So they're no longer now leaf cells or petal cells or root cells. They're all just general plant cells. And this lump of cells, now that they've de-differentiated, is called a callus, what we spoke about earlier. It's usually white in color. So you can see, you can take yourself through this process. It's again another way of showing the plant tissue culture. And again, after we form the callus, the sterile conditions we know are essential because if there are bacteria or fungi landing on the agar plates, they'll obviously grow very rapidly because of this nutrient-rich medium, and they'll infect the plant tissue and take over the entire medium. You can see here some pictures where bacteria or fungi has grown within the medium. These are the sterile conditions in the plant tissue culture lab. Then the explant is left to de-differentiate, as we said, in a growth chamber. This growth chamber needs the correct light and temperature conditions, which encourages the growth and division of the callus. Once the callus has developed, it can be separated into a number of callus lines that can be kept permanently for use. So we can just use these calluses to continually produce new plants. We can then genetically modify the callus cells by exposing them either to recombinant bacteria or shooting them with a gene gun. So what a recombinant bacteria is that in a bacteria cell, we have something called a plasmid, which is another type of DNA within the bacteria cell. It's a circular DNA. Scientists can clip this circular DNA and insert a gene that they want to introduce to the plant. The plasmid then reforms and is inserted back into the bacteria, and then the recombinant bacteria, recombinant means it's been modified, is then inserted into the plant cell. Or the callus cells are genetically modified by being shot with a gene gun, where tiny pellets that are covered in the genes, the desirable genes for the cell, are shot into the plant. So as I said, this bacteria can genetically vary the DNA of a plant naturally. It does it itself. Humans have harnessed this power and used it to introduce new DNA, the desirable DNA, into the plant. The problem is that there's no control over where in the genome the new gene is inserted and how many times it's inserted by the plasmid. When we genetically modify a plant, we now call it a transgenic plant. So as you can see here, this would be the bacteria cell with the plasmid that's been genetically modified. It now goes into the plant cell. It becomes a callus, and then it's given hormones to go back into the plant form, forming a plantlet and then a plant, which is obviously transgenic because it's been genetically modified. This is the recombinant bacterium method. This is the gene gun method. You can see here the process of a plant cell becoming a callus. Once the callus cells have been genetically modified, they don't always have to be genetically modified, but most of the time they are. They're then placed in a different agar medium containing a different combinations of hormones, and these hormones encourage the differentiation now of the callus into embryo cells, which causes the growth of roots and shoots. So we've now formed the callus. Then the callus is placed in a different agar medium with hormones that encourage the formation of an embryo. The embryo then grows roots and shoots and eventually becomes an entirely new plant. Here is a picture of differentiated cells developing from the callus. The plantlets are then kept once again in a growth chamber where things like light, wind, humidity, and temperature are controlled until the plantlets are large enough to be planted outside in irregular soil. It's often necessary to expose these plantlets to natural conditions slowly in order to prevent them from dying from shock, where they go suddenly from this strictly regulated growth chamber into a completely non-sterile natural environment. And what we call this process where you introduce it slowly, we call this hardening off. Here's just a picture of a scientist. We can see this is the process of plant tissue culture, where we take a tissue sample from the plant, place it in an agar medium, it de-differentiates, it then goes into a different agar medium, and it grows into plantlets and then into plants itself. Where you can use any plant to create the callus, obviously because of the totipotent nature of plants. How does asexual reproduction benefit future crop production? There are a few possible benefits to asexual reproduction. Obviously, propagation is faster, so reproducing these new plants is a lot faster than from sexual reproduction because there's no gamete formation, pollination, or fertilization. All the propagated plants are of a consistent superior quality, so they all have these desirable traits, and we can produce large quantities of plants cheaply, easily, and quickly using cuttings, bulbs, tubers, grafting, etc. Using the graft technique, fruit trees will grow more quickly to maturity and this to fruit production because you choose the plants that with qualities that make it grow more quickly, that make it produce more plants, etc. And by micropropagation, numerous new identical plants called clones are produced. Some more advantages is that we often choose micropropagation over traditional crop breeding because micropropagation allows plants to be mass propagated in a short period of time, as opposed to sexual reproduction or traditional breeding. Obviously, micropropagation usually ensures disease-free plants because you choose the cells that are free of disease and culture them in sterile conditions where they can't get a disease. You can propagate all year round. You don't need to rely on seasonal changes, such as plants that will only grow in certain seasons. And you can use micropropagation in conjunction with genetic engineering to propagate transgenic plants from genetically modified cells. So you can genetically engineer the cells into plants that you want with desirable traits. So those are the advantages of asexual reproduction. one of the things that's important to know, they'll sometimes ask you about GMOs, which are genetically modified organisms. And it's sometimes a controversial issue because people don't want to eat GMOs because they believe that they've been contaminated, or the genes inserted into the GMOs might be eaten by the person, and then the person will get these genes. And just the counter argument to that is obviously science doesn't support this evidence. You can't eat a genetically modified organism and then get the gene. You're not going to suddenly get this plant gene that makes you produce apples or something. So it's not scientifically evidenced that GMOs have any adverse effects on the human body.